to spectral phasor. Give me a second. I will like to basically move this and start my countdown. So about spectral phasor for hyperspectral imaging. And I always like to say when I begin this talk that this started when I was here in Uruguay doing my PhD, and one day I met with Enrico and he said, well, Leonel, you are able to do spectral phasor. And I say, no, we cannot do phasors in Uruguay. We don't have any lifetime machine. I say, no, Leonel, of course you can do it. You have a spectral machine. And I say, what these have in common? What are you talking about, Enrico? So after that, he explained me how to do spectral phasor and how to use this unique tool to analyze hyperspectral imaging. And we published several papers that I will cover today. So trying to answer in the question about what are the similarities between lifetime and spectral phasor is, well, as Enrico say and Dave say today, well, if we have lifetime decays, we will find single exponential on the universal cycle. If we increase the lifetime, the modulation will be decreased and the phase will be increased. And if we think about basically on spectra, the main concept here is the period. So as Enrico said before, we need to have a period, a fixed period, in order to transform our data and basically follow all the rules related to phasors, linearity, linear combination, etc. So, but if we do the proper transformation, and I will address this in the next slide, we can have a transformation. Now it's not just 1 quarter, it's the 4-quarter phasor transformation. And we will have a modulation and a phase, and I will explain in a minute what this means in terms of the characteristic of the spectra that we have here. If the phasor is the spectral moves, this will produce a shift in the spectral position and change in modulation and change in phase. So in some way, they are very similar. The big difference is that we don't have a universal cycle. The universal cycle is this one, but it doesn't have the meaning that we have here. But still, all the properties that we have learned from the lifetime phasor will work in the spectral phasor. So about the formalities, well, this is a Fourier transformation. As Enrico said, we will get basically the real and imaginary part of the Fourier transformation that we call GNS. And we have basically the integral of the intensity multiplied by the cosine or the sine of the shift, spectral shift. This term basically defines how much is shift from the beginning of our range. And we always define a range. And this is, as I said before, important. The other very important thing is that we need to normalize. Without normalization, all the linear properties will not work. And this is important. What are the advantages, or what are the highlights that we have with the spectral phasor? Well, it's a model-free, as Enrico said, does not require previous knowledge, and we will see that this is very important for some of the application. It can be done in cuvette and imaging, and it combines the classical analysis of the mixing with the new phasor analysis. And it's very, very important, this application, when we use environmental sensitive proof. So this method was originally described by Freduoni at the Harrison lab, and the main interest at that paper was the demixing. And this is what I will explain now with this simulation that I did. So if we simulate Gaussian curves like this, that we have different width and different maxima, and then we transform our data to the phasor, you may notice that we have points in the phasor that are spread around of our phasor plot. Well, the location, so how far are from the center, is related to the modulation. And the modulation means that as narrow is the spectra, more closer toward to the perimeter we will be. On the other words, we can say as broader, closer to the center. So the next thing to notice is that if we shift from blue to the red, we will change the phase starting from this point. So just looking at the phasor, I can immediately tell you that this is bluer than this spectra. And I can tell you that this spectra is broader than this spectra. Not even looking at the data, just looking at the position of the phasor. So this is really, really important because I don't need to include in my analysis any further information. So as I say here, the width and the center of mass are the two main important characteristics to make the spectral phasor work. And basically, one of the big properties is the linear combination. So if we have a mixture of any of these two components, the point will be on the line that join the 2 points. And if we have 3 components, of course, we will have a position within this triangle. And today we will see a very nice new developed by Alec Bagmishana that they can do not only 3, but even more, 4 and many more if you can find or define the positions, but also if you don't know the position. And this is really, really important. The other property which is important is, well, as Enrico said, the reciprocity principle, which means that if we have an image, I represent here an image, each of the pixels here is an spectra, and one is the addition of all the spectra. And if we do that, and we place cursors in our phasor, we will highlight the image from the pixel from where the spectra come from. And this is really important for images analysis because without any knowledge, you can immediately find where are the image, what are those pixels in the phasor and the image. But it's also true the other way around. So if you want to select small region on your image, you will isolate where are those pixels in this scatter plot. So summarizing this, well, one of the most important is the model-free analysis. And of course, if we go to higher harmonics in the Fourier transformation, we will have better resolution, but we will have to take care because we can have more revolution that can basically overlap the pixels from different wavelengths. So which are the instruments that we have in the market and which are the development that enable us to do phasor? Well, there are different brands, like Zeiss has the 780, 880, and the 980 with truly spectral imaging, 32 channels, parallel, which is similar to this array that I show here. And this is the example. They use different kind of optics to produce basically the opening of the spectra, diffraction gratings, or also prisma, and different configuration that you can project all the light on the array, or you can take the spectra one by one, moving the prism and selecting with your PNT one of the band that are passing through. So this is, for example, the case of Leica. Nikon has also a truly 32 channels. Olympus use this other strategy, basically in the moving the slit. And there are also the spectrographs coupled with a camera that you can have really high resolution to nanometer bandwidth. But of course, this needs longer integration. There are some other base camera like the one that Enrico has in his lab with the Andor and Zion MCCD, which is really, really good. And more recently, there is a way to do spectral phasors and hyperspectral imaging without having basically the spectra and without having a spectral detector, which is the sine cosine filter. And we will have I will explain a little bit at the end. And I think tomorrow Enrico will talk about Diver and he will address this again. And today we will have a talk by Nick Hede about the sine cosine and the light sheet microscopy. Okay, how this look like? Well, basically this is taken in the SP5 that we have here. We used to have here at the Institut Pasteur. And you may see here how it changed the maxima along. I am getting the different image from the range. I will do it running again. These are cells with these droplets, which are basically lamellar bodies. And they are labeled with a solvatic chromic proof, Actan. And then if we do the transformation from each of the pixels, we get our cloud of pixels. Each of these points are one of the pixels in the image. and then we can use a color design, and then you may see that now that this is basically an homogeneous intensity, is now rather a completely different organization, and you may see immediately those very noticeable lamellar bodies which are completely different from the others, and these are very blue. The purple here means basically shorter wavelength, and the red one are longer wavelength by the phase. Zeiss does this in a different strategy. As I said before, they took 32 images, and then they basically have the spectra pixel by pixel, and they can color the spectra based on this scale, which is assigned to the maximum. Where is the maximum of the spectra along all the 32 images? So what are the considerations that we need to have when we do the spectral phasor? One of the first one that I want to highlight is the range. And what happened with the range? I mean, you may think that eventually, if you increase the number of steps within a period, you will increase the resolution of the phasor. And this is not really correct. And you will sacrifice something which is very important for the phasor, as I will show you in a minute. Well, if we do this, for example, these are the spectra for night red in different environments, so phospholipids or triacylglycerols. And we have one which is very, very blue, and the other is a little bit redder, and this is the total. Then we have the phasor. And you immediately notice that this point, which is basically the addition of this spectra and this spectra, is mathematically added. I took this spectra from the webpage of Thermoscientific, and I just sum both. And this should fall basically in the middle of this linear combination. And you can immediately notice that this is out of the linear combination. And this is because you cut almost half of the decay here and almost 30% of the decay here. And this is one mistake that you make do if you don't know that the integral of the intensity is very, very important. So if you collect the total spectra and you allow to relax the full spectra in the two sizes, everything came absolutely in the line and everything worked perfect. So the other is, well, what happened with saturation? Well, you immediately will notice that if you saturate it, the spectra will be distorted because you will chop some of the channels, and after that, the center of mass will be moved. Well, I did this simple experiment, increasing the light over and over and over, and I placed the cursor at the beginning in some location, and you may see that as soon as I start to increase over and over and over, and the saturation go up and up and up, this cloud of pixels move away from the original position where everything was okay. So as a recommendation, please take care of the histogram, and always took your data looking at the histogram and try to avoid to go to higher intensities. So the second thing is the consideration of the channel and the bandwidth of the channel. You may notice you can have, if you have an instrument with 32 channels, this is ideal because you got the 32 channels in one single shot, but sometimes you need to record one by one. And you may think that eventually if you collect 8 or 10 or 16, you may save some time, because every time that you illuminate is more bleaching, is also more time, so the things can move, anything. So anyway, so of course, if you can get 32, is almost ideal. If you go higher, indeed, you renounce, because of course, in each of the channel you have LED signal, and this make the spectra broader, the spectral phasor broader. So between 16 and 32 is kind of the ideal situation for the spectral. And if you are lucky and you have in your lab the 32 channels, don't move anything, use this configuration, which is really, really good for all the analysis that we will see in the presentation. So to explain the properties and the goodness of the spectral phasor for the analysis, I will use this, some of the experiments that I have done during my PhD and also the postdoc, working with solvatochromic proof, Agdan and Laurdan, original synthesized, as Dave said yesterday, by Gregorio Weber and Faye Ferris. Basically, this has really a unique photophysics. After excitation, depending on the environment, the fluorophore can basically emit fluorescence without much change. But if you have solvatochromic, if you have dipolar molecule around, this dipolar molecule can be reoriented by the energy of the dipole of the probe, and this energy will be reflected as a reduction in the energy, which means a shift in the spectra and also a change in the lifetime. And this is basically what we have here in this analysis of the polarity of liposomes with Lardan. And you may see that if we increase the temperature over and over, we change the spectra a lot. How we use, well, basically, I want to stress this concept here. What we are looking basically here is just as at this molecule of water which are in the close proximity of Lardan. And this water molecule will be affected by the physical state of the membrane. So if we have liquid disorder, shell phasor, or liquid order when we add cholesterol, this will affect the number of this molecule, and at the end, the emission spectra and the lifetime of the Lardan, and this is our readout. So traditional, this was analyzed with a method developed by Enrico and Tiziana Parasasi that was known as generalized portalization, and the name generalized means that you don't need to use polarizer, as Dave talked yesterday. We just use basically two bandpass filter, and we use the polarization function. And the reason for why we use the polarization function, because it's linear and you can it's additive, and you can analyze component. This can be done in a cuvette, and you collect only one intensity, or in a microscope, collecting two bandpass filter. And this was really an amazing work done by Luis Bagatoli on the early 2000s, using this in a microscope when he was postdoc with Enrico, and many more then follow this strategy, and today is very, very popular. But what are the problems here? Well, we start from a model of two components. So we define two components. And so Enrico say, well, with this kind of thing, sometimes it's not good just to basically guess from beginning that we have only a shell or only a weakening a liquid phase, and then we have something in it. What happens if we have, I don't know, autofluorescent, or if we have a third component or something? Well, this cannot be, you cannot easily put in the model basically a third component in the GP analysis. So this is the first example. I made vesicles made of DOPC and DPPC. For those of you who are not in the membrane field, DOPC is a fluid membrane at the temperature that we run the experiment. DPPC is a solid membrane, and basically the mixture of both 50-50 will give you a coexistent on fluid and shell membrane within our system. So if we took this and we calculate the GP, we get a point. Okay, perfect. This is the point, very high GP. Then we can start adding cholesterol and make more complex the system, because after certain amount of cholesterol, we will have liquid order. So in some situation, we will move completely to liquid order, or we will have liquid order fluid or the three components. So if we transformate our data, we get this list of values of GP, and this can be eventually analyzed for a complex model. This is not a fitting, this is just a draw that I did on top just to illustrate, but basically we need to propose a model to understand what is going on here. But if we transformate our data to phasor, look what happened. Well, we are going from this point to the other point, and everything comes in a line. So which means that we are looking at these molecules of water that we said before, and everything is a linear combination between the different states of the membrane. So the next experiment that I want to show is related to imaging. And this was a part of my PhD, and I was interested in the organization of this intracellular organelle, which is called lamellar body. This is located at the lung in the pneumocystis type 2 cells, and it accumulated. It stored the pulmonary surfactant, which is a substance that we need through the breath. So what we know about the organization and the physical state of the membrane of this organelle on the process of the maduration is by the electron microscopy. And we know that there are different steps, the multilamellar vesicles, the composite body, the lamellar body with different stages. And then when you are, the lamellar body is mature, the lamellar body will release all the content in the interface of the alveolus, and it will work as a tensioactive substance. Well, we were working at this point with this experiment, and a paper came out saying that the lamellar bodies are very high dehydrated and packed substance within in vivo cell, and they assign this to a shell phase within the pneumocyte type 2 primary culture. Well, this was very, very bad for us because it was truly, I think, 15 days before we submit our paper. But the good thing was that we were doing spectral phasors. And we noticed something that was very, very interesting. So basically, we plate these cells with produced lamellar bodies, and then we wait until confluence, and then we wait until different times, and then we analyze basically the organization and the size of the lamellar bodies. So if we do that, and the first thing that we analyzed was structural fixtures. So we measure the diameter, and we notice that the diameter increased over the time, which is expected. If you look at the distribution of the sizes, this has more information because the histogram are not always with the same width and distribution and the same center, which is something interesting. But then we analyzed the spectral shift that Laurent suffer when the lamellar body are going from day 1 after post-confluence to the day 14. And you may notice that, of course, lamellar body growth, but also it changed the color. Means that you are moving from pixel within this cursor, blue, to red, green, and then to red. And you see that this is a linear combination. So in the middle are basically combination between the two ends of the different stage of the lamellar bodies. Well, this alert us immediately. And when we did this analysis, which was, okay, let's compare the size of the lamellar body as a diameter versus the number of pixels. So in some way, we are trying to control the fact that as bigger are the lamellar bodies, more bluer they will go. We found this very interesting profile. So for the first 7 days, there is not much change in the spectral phasor position. But after this point, there is really a huge increase. And this is also true for the cursor in the center, which is, in a way, the increasing of the blue point as a linear combination of blue and red. Well, what happened, as I said before, the different stages of the maduration of the lamellar body takes first to go from the multivesicular to composite body, then early lamellar body to mature lamellar bodies, and then to the final stage when we got all the lamellar body mature and with very compacted and unrelaxed environment. What we think is happening is that until the lamellar body gets certain size, they are not starting to fill up with the phospholipids. And this is, we think, associated to more or less 7 days. Okay, but we have phasors, and phasors enable us to do something more, which is this thing? Well, if we analyze the position for known phases like DOPC as a fluid, DPPC as a shell, and then a liquid order phase made of DPPC plus 30% of cholesterol. And we pick this 30% because the lamellar bodies within the cell does not have this higher concentration of cholesterol. They are below 10% of cholesterol. Then you may see that we have some sort of triangle here. And if we have our lamellar bodies with a membrane phase, which is any of these linear combinations, well, the lamellar body should be on this linear combination, any of those. So if the lamellar body were held shell phases, as this paper showed before, it should be somewhere here or there with different fraction of fluid, whatever. So this data here between cyan and green are the data for the I549 cells. And basically the lamellar bodies are exactly in this cursor. So they are out of the linear combination between liquid order and fluid membrane. And we immediately thought, well, what is going on here? These people say that this is the lamellar body should be shell phases. Well, we did an experiment, which was, okay, let's analyze the lamellar bodies after we secrete and we collect from the extracellular environment. We couldn't believe what happened, but what happened was this was the original position. The lamellar body, after they are moved, they are outside the cells, they move to the linear combination between liquid order and fluid. And what is this? Well, what we believe that's happening is, of course, the intracellular environment is completely different to the extracellular environment in terms of water. And water is really an active molecule to modify the structure of the membrane and produce mainly change in phase transition, not only in the liquid order disorder, also in order dimension, like, for example, inverted hexagonal cubical phases. And what we believe is what this is happening. In the interior time of the lamellar body, everything is in another phase. We really don't know, but we basically are speculating with this. And when they get outside, which is full of water, the lamellar body change completely. Okay, so I will go over this because this is more biological, that really doesn't matter. And I will go to this thing. During the dealing with the reviewer, we have to demonstrate that these were lamellar bodies, because can be also, I don't know, lipid droplets. So I got a plasmid with a molecular marker for the lamellar body. Unfortunately for me, it was green. And this is the linear combination of the Laurdan, and this is the position for the green fluorescent protein in the spectral phasor. And if you place basically Laurdan plus green fluorescent protein in the lamellar bodies, you may see that these are the cursors with different linear combination of green fluorescent protein and Laurdan, and these are the cells. Unfortunately for us, we cannot see which of the phases are the lamellar bodies. So, well, I thought, well, there is something that I can do. Okay. At that time, I was really completely out of the molecular genetics, and I was not able to do any genetical conversion between one protein to another. And for me, the simpler way to address this is through biophysics. So I basically thought, okay, if I can find another proof that is in the other side of my molecule reporter, and I can prove that this basically molecule works more or less in the same way that Laurdan, okay, I will be able to demonstrate this. And this is exactly what I did. I compared Laurdan with Nile red, and if you co-label the cells with Laurdan and Nile red, you may see that basically what we have is a linear combination of the unrelaxed Laurdan with the unrelaxed Nile red, and then a linear combination of the relaxed Laurdan with the relaxed Nile red. This proves that basically this work more or less similar, and now we are able to do our analysis with the lamellar bodies. Okay, this is what we did, and you can immediately see that the linear combination is coming from the places where our lamellar bodies are, and we basically demonstrate that these were lamellar bodies, and we demonstrate that there are other structures which are with different environment, which does not require to be in the lamellar bodies, and the problem was solved and the paper was accepted. I want to make a point here, which is very important in the comparison with HEP. You may see that we can keep working with the information of Nile red even when we have a third component, which is the green fluorescent protein. And we can still assign this to the phase that the Nile red is. And this is very important. This is not that simple to do with the HEP. It's almost impossible to do with the HEP. And this is a big point with the spectral phasor. Okay, then I want to move to the combination of spectral phasor with lifetime phasor. And this was something that Enrico put on my front when I just joined the LFD. And we did this set of experiments to demonstrate that this is possible. And the rationale here is simple. Okay, if we can get two images, ideally simultaneously, but if not, just one after another, and then we do the transformation, the image is common for both. So we have a common thing between the spectral phasor and the lifetime phasor. And the reciprocity principle enables us to highlight a region in the spectral phasor, look where those pixels are in our common image, and then find those pixels on the lifetime phasor. This is a unique possibility that phasor enabled, and the linear combination of the phasor enable us to do, and it's very useful, and as we'll show today later in the afternoon, to analyze really complex photophysics that can happen in the cell. So we proved that this worked. We did this experiment with cells, and you may see that if you highlight the blue pixel in the spectra, these are the longer lifetime, which is what we expect. And then if you highlight the shorter, the longer wavelength, will give you the shorter lifetime here, and then the linear combination will be in the middle. So everything works. You may see that this is a little bit scattered, but if you look at different masks, you may immediately notice that the linear combination came more soft and more narrow, and this is because we are analyzing a specific region of the cell, like for example, the plasma membrane and the intracellular of the cell. So finally, I think I am very good on time. I will explain a little with this is, okay, can we do a hyperspectral imaging without having an hyperspectral detector or a machine that can collect the spectra? Well, the first time that I hear this from Enrico, I thought this was really esoteric, but it works, and it's unbelievable, because what we can do here is the following thing. Imagine that now we can have a filter in where we can put a mathematical function. The mathematical function is a sine or a cosine function. I thought this was unbelievable. How you can put a mathematical function within an optical object? Well, it's not a mathematical function. It's a transmission filter that has a shape which remains a sine or cosine. These are ideal sine and cosine filter, and these are the real sine and cosine filter that we use in our experiment. If you remember, in the transformation, we multiply our intensity by the cosine or sine of the spectral shift. Well, if you think about it, this is exactly the same. We are integrated all the bands from a period multiplying by a function, which is the transmission filter, in the range. If you can collect the two filters plus the total intensity, and you apply the proper mathematics, you can get the Chi and the S, and at the end, the spectral phasor. Well, we did that. Indeed, Sasha Dvornikov and Enrico did this in the original paper in Biomedical Optics in 2018. And then we proved with other examples, and I picked this from a paper that we published with Sasha together, in which we originally demonstrated with beads, orange bead and green bead. So if we do that, and then we transform it to the phasor, well, you can immediately notice that we have a cloud for the green and a cloud from the orange, completely split, and there are few pixels. And this is really nice point to make. Enrico said that you can see few pixels. I will say you can see only one pixel in which you have 50-50%. And this is unbelievable. You can see one pixel, only one pixel which is in the middle, and where, for example, the point effect function cannot resolve these two things. Then we prove this with a more complex sample, and these are bonds, and the bonds, you can image the lifetime of the collagen 1 or the collagen 3, and we know, and Sum and Ranjit will show today how this can be used for cells and for tissues and other stuff, that the lifetime change, and if you select with the pixel side, with the cursor pixel here, you highlight the pixel in one and the other, but when you do spectral phasor, you can achieve the same. The spectral shift between the two collagen enable you to do the separation between the two regions. So finally, I want to emphasize and make some points about how good this spectral phasor can be for analysis fingerprinting without performing any fitting or without assuming any model. You can do the mixing. You will see really much better this on the Alex talk today. The linear combination are very simple to follow, and it's really intuitive work. We demonstrate how valuable it is for membrane studies. We can do a very nice conclusion about our system thanks to the spectral phasor. And then we demonstrate the value of the multidimensional phasor, and more recently, with a single detector, we can do also spectral phasor using the sine cosine. And to finalize, what I would like to do is to make a point on some remarkable things that are going on in the recent time. And the first one is related to one of the attending, Ernesto Ambrogio, that he showed how valuable it is for the autofluorescence of tryptophan binding. These are construction of GAG protein binding to liposomes. And if he has different construction with methylation and without methylation, this follow different interaction with the membrane. And only the spectral phasor enable them to show that there are really some kind of interaction when they don't have the myristoyl and they have a completely different and really more complex, which means that you have three component analysis. And I found this application very, very nice for the cuvette community and for the protein community. Then tomorrow, Lorenzo will speak about the combination of lifetime and spectral due to the spectral phasor, but in a truly time-resolved lifetime machine, which is, well, time spectral resolved lifetime instrument, which is unbelievable. And today, Nick Hede will address this application of the sine cosine for the spectral analysis in a light shield microscope, which is also unique. So thank you very much for all the collaborators, Enrico, Dave. The lamellar body was done with Luis Bagatoli and Soledad Estrada, and we did a lot of stuff together with Sasha Dvornikov. And of course, I will be more than happy to answer any question from the audience.